good afternoon, and it's great to see everyone still continuing here at the workshop. I'm Joe Monaco. If you don't know me, I work in the Office of the Brain Director, and I'm, as Young said earlier today, I work with her as a co-lead for our data programs within the Brain Initiative. So I'll be your MC for this session. And so first up, we're focusing in the session on AI-enabled brain cell atlas knowledge bases, continuing that discussion about how to leverage foundation models and agentic AI to transform brain atlas data into these intelligent, queryable resources that we're all envisioning to accelerate scientific discovery. So going into the keynote here, I'd like to introduce you to Dr. Jure Leskovec, who will set the stage for our panel discussion with his presentation. So Dr. Leskovec is a professor of computer science at Stanford University. He's affiliated with the Stanford AI Lab, its machine learning group, and the Center for Research on Foundation Models. He pioneered the field of graph neural networks and co-authored the Python library called PyG. His research spans social networks, data mining, and machine learning, as well as computational biomedicine. So work from his group has been used to fight the COVID-19 pandemic and has been incorporated into industry products. So his presentation today is Foundation Models and Agentic AI, Building the Intelligent Infrastructure for Brain Cell Atlas. And at this point, I will turn it over to him.
Yeah, thank you so much for having me. Thank you for the opportunity, and also thank you, Joseph, for a nice introduction. I assume everyone can hear me and everyone can see my screen. What I want to do in this 20 minutes is show kind of and propose the direction of how AI can help us make progress in building this brain cell atlases and kind of building the understanding, the intelligent infrastructure on top of brain cell atlases. And my talk will have kind of two main points. One will be about building these large-scale pre-trained models that really allow where the biology emerges right out of from the models and allow us then to do a lot of super cool stuff. And then I'll also touch base about the agents and the age of agents as interfaces through which human scientists can use these advanced tools, these advanced models to accelerate science. So first, I want to talk about these large-scale pre-trained models, which we call foundation models. And we have foundation models for a number of different domains and disciplines. One examples of foundation models kind of for general use are large language models. We have foundation models that understand computer code really well. And of course, the use of foundation models in biology, in biological, biomedical domain is also emerging. So now you can say, how is this different from what we know and what we've been doing, working on in the past? And maybe the way I would define this is that a foundation model is a model trained on a massive amount of unlabeled data. So it's just kind of trained on raw data. No human labels, no cleaning, no supervision is given. And in this respect, whatever emerges out of this model that is trained on this unlabeled data is truly emergent, right? Kind of the biology, the organization, the understanding kind of emerges out of these models. And the beautiful thing is now that this type of representations that come out of these models can be adapted to many tasks, to many types of analysis, and allow us to do more accurate, let's say, prediction analysis in the future. And the way this is fundamentally different from kind of classical machine learning models is that classical models are usually task specific. They require labeled data. Somebody needs to develop them, and so on. So based on this exciting idea, we have published an opinion piece with a number of collaborators from Chen Zuckerberg, BioHub, and elsewhere about this concept of an AI virtual cell. And one of the key concepts there was that if we truly would like to have this concept of a virtual cell, we need to model biology across the scales, right? We have to model it from molecular scale, understanding the molecules that are in the cell, at what locations, positions they are, and who they are. We need to then be able to model individual cells that are, let's say, collections of these molecules. But actually, we need to go even a physical scale higher up to the multicellular scale. Where we may be able to want to model entire tissues, their organizations, or entire patients. But we want these models to be informed all the way down with the individual molecules that we find in these patients. And we have proposed a blueprint of how we can build basically large pre-trained neural networks, how we can connect them, that allow us to do this type of modeling, basically molecular scale informed modeling of, let's say, entire patients. And the interesting thing is if this type of ability for us is there, if we are able to create this type of large pre-trained models, then basically this means we are able to model a number of different processes on top of these types of digital twins, let's say, of cells or of tissues or of entire patients. So the point is that through this, one amazing thing we get through this type of pre-trained models is that we get this one reference space, a reference space where all the biological data, all the cell, let's say single cell data or the patients can all be mapped, and we can now reason about them in this single representation space. And the point about this single representation space is that it's unified. It does not require us to train a separate model per cell, per organism, per experiment, but it's a pre-trained model that just maps all the data into the same space. So what does this mean? Is that now we are able to build additional tools, we call them virtual instruments, on top of this unified representation. Right, so we can take, different reference cell atlases and kind of map them into this space. So now that every additional experiment we do is now mapped and understood in the context of the reference cell atlas. We can think about understanding the locations of, let's say, different cells, in this case, to discover spatial niches, to discover novel cell states, and things like that. We can also think about perturbation type modeling and dynamics of how the cell state is changing over time, again situated inside this single reference space in which we also can kind of map other reference atlases. We can think about modeling perturbations that are basically now just in some sense saying how does a cell move from one part to the space to another part. We can model intrinsic perturbation, we can model extrinsic perturbation, because we have this now unified space in which, let's say, cells can be represented. And that's really the kind of the key benefit here, is that we have now one unified representation space in which all these objects, entities, cells, for example, from different experiments, tissues, donors, and so on, can be mapped, and we can reason about them in a unified way. So we have set on actually a path to start building this layering of foundation models, to think about foundation models at the molecular scale, at the cellular scale, as well as at the multicellular scale. And at the molecular scale, we already have some of these models built, like genomic language models, protein language models, and so on. These would be examples of these, like EVO, ESM, and so on. At the cellular scale, we can now say, let's build models that are actually informed by the molecular scale representations that are of the molecules that are found in the cell. So one example of the model we have built at this level is called universal cell embeddings model, that actually is using the individual, for every gene, we are using the embeddings of the proteins that those genes are coding for. And then we can also think about building models at the multicellular scale, understanding the spatial organization of the cells through graph-based analysis and so on. So what I want to show you next is give you a few examples and a few vignettes of how these models can be built and what kind of interesting new capabilities they allow us to do. So first I want to talk about a model we call universal cell embeddings model. The interesting thing of this model is that a single pre-trained model that can create an embedding, a representation of any cell coming from any organism. And the way the model is built is that the input to the model is single cell RNA-seq data of a cell from a given organism, as well as the individual proteins that the genes in that organism are coding for. So now we are basically representing genes by the embeddings coming from the cellular, from the molecular foundation model that is encoding the structure of those proteins. And what this means is that now a single model that is pre-trained without any human supervision allows us to take, let's say, data from multiple different organisms and all map it into a single reference space without any batch correction, any alignment. None of that is needed. Just we raw data and we obtain these embeddings. Here I'm showing you data from 3 different organisms, fly, mouse, and human, and how, you know, kind of clusters emerge and how the data is well integrated. And what we have actually done as a part of our collaboration on the Brain Explorer project that's funded by NIH, and it's a collaboration between University of Santa Cruz and UCSF and Stanford, is that we were saying, okay, can we build a brain single cell transcriptome kind of reference atlas? So we went and collected a number of different data sets, about 20 million cells from human nervous system cells from 92 different studies. And here I'm showing some of this, and you know, this work was really done by Jing Zhu from UCSF using this. And then what she did is she took these cells and she used the universal cell embeddings model, the UCE model, that has never seen this data set, has never seen these cells, these studies, these sequencing technologies, and so on, and just said, let's embed this and let's see what structure emerges. And here I'm showing you the structure of the embedding space where the cells are colored based on the data set they are coming from, and you see this nice integration, emergence of clusters, and so on. We have then made further progress where, for example, we can now take 3 different new data sets, the BICAN basal ganglia data set, the Gabito et al. Data set, and Silete et al. Data set. And again, take these 3 data sets, around 7 million cells, and just embed them into this shared space. Again, this is all done with a frozen model. Nothing is trained. We are just using a pre-existing model that has never seen these data sets or these cells or anything like that, right? And we are embedding the raw data. And again, you see very good integration across these data sets. Here, we are coloring the cells according to the data set they are coming from. You see emergence of these clusters and so on. And of course, you can be asking, okay, are these clusters meaningful? So what I'm showing here on the next slide is basically the same UMAP, the same integration, but now the colors are different because the colors are according to the cell types as were annotated by these different studies. And again, you see that now these clusters have cells of the same color, which basically means that the notion of a cell type just emerged out of this model, even though the model was never even told what a cell type is, was never trained on any cell type labels or anything like that, was just trained on raw gene expression vectors, right? Again, with very small amounts of brain data or anything like that, right? But we are seeing how this structure is emerging. What is interesting now that we can basically use this, let's say, this set of cells as our background reference cell atlas, and we can take a new data set. Here I'm taking a data set from a 2023 study from Jorstad and all, where you can now see how the cells from that study are mapping on top of my reference atlas, and I'm showing this in black, and you see kind of how super nicely it fits over the existing clusters and over the existing data. And again, all this with a frozen pre-trained model that has never seen this data, has never been told what cell types appear in neuroscience and so on. So this becomes quite interesting, right? Capability, because now we can basically take any data set or any set of data sets, and we can kind of map it on top of reference data, all independently processed within individual studies, and just send them through this neural network that is basically, in some sense, on the fly, doing alignment, doing batch correction, and things like that. And this really now is a new capability because allows us kind of to situate any cell from any experiment in the context of the let's say richly labeled, richly verified reference, right? And as we make progress, we can further improve that reference. We can understand where the data is coming from. We can label it and so on. But this means that any new experiment can naturally kind of map and add into the same reference space. So that's really kind of the core of what I wanted to communicate today. What I'm also going to show you is that this idea of going from molecules to cells to multicellular representations actually works. So we have also been working on a method called Pulsar that allows us to create multicellular foundation model, where basically in this specific application, we can take a set of single-cell RNA-seq profiles from a drop of blood from a patient, and we can now create a patient-level embedding that is informed all the way down to the individual gene expression levels at each individual cell of that sample. And we can train, again, this model in a fully unsupervised way, no human labels necessary. And now we can obtain patient-level embeddings, right? So you can basically take any set of single-cell RNA-seq cells from the patient, and you can now embed them into this unified space. And what is interesting is, for example, that clusters that start emerging in this space are kind of clustered by the disease, even though this disease information was never used and never at the time of training the model. So just kind of showing that this vision of building these unified spaces in which data can be mapped, and the kind of the organization of this is fully self-emergent. And then in the last few minutes, what I want to also talk about quickly is about how agents change all this, right? And we really think of agents as kind of about empowering human scientists and accelerating the rate of science. And what we've done in my lab is we built Biomni, which is a generalist AI agent used by over 15,000 scientists all over the world. Right, the idea here is that we want to empower every scientist. We want to augment the biologist with the power of AI. To basically every biologist now has an entire team of experts at their disposal. These agents are universally accessible, dramatically reducing barriers and amplifying kind of the potential of human scientists. What's an agent? An agent is a piece of code that has access to tools to take actions in some environment to, let's say, perform analysis, perform experiments. Our Biomni agent has access to over 250 different tools, databases, sub-agents, and can use them to solve tasks that are given by the human, right? So there is PDB, there is ScamPy, there is AlphaFold, there is BioPython, there are all kinds of AI models, browsers, lab protocols. All this is at the disposal of the agent to basically use these tools to perform analysis for the human scientists. This tool is openly accessible, can do a lot of different tasks, from single-cell RNA-seq data annotation to GWAS studies, to drug repurposing, to rare disease diagnosis, and so on. We've deployed it with a number of different labs at Stanford, for example, for a wet lab experiment protocol design, for analysis of omics data combined with wearable sensors data from the Snyder lab here at Stanford. And we made it freely available for a number of different users. As I said, 15,000 now who are essentially using it daily. What we see is that the speed of science is accelerating by 10x, right? A scientist with this type of agents can just execute tasks, use these tools in a much more natural and easier way than, downloading code, setting up Python environments, and things like that. So where does this leave us? To conclude, right, is really the future, how we are going to accelerate sciences by use of AI, either through this virtual cell or virtual digital twin models that are pre-trained on large amounts of data, as well as with agents that sits on top of these complex tools, are able to use them and orchestrate to speed up the analysis. Of course, the time is early. There is a huge amount of work that is still open, many open questions around auditability, tracing, hallucinations. But even though these tools are very imperfect, we see huge value for them and huge appetite among the scientists to be using them. So with this, I'm done and very excited about the panel and the questions. Thank you so much.
Right. Thank you, Dr. Leskovec, for that overview of agentic AI and foundation models as applied to all the things that we're talking about here. We're a minute or two behind, but if we have probably time for one question or maybe a couple quick ones, but
So I have a question. So basically, when you say that you feed the program raw RNA-seq data, so I mean sometimes the data set is quite large, and usually when people do cell type analysis, they look for specific marker genes to group these cell types together. So are you just feeding in the raw RNA-seq data, or are you looking for specific genes, like marker genes, when you feed it into this program?
That's a great question. Thank you. So we are feeding in single cell RNA-seq counts.
So how do you how does the program handle that level of data? Because it's like, it's very complex data, and not every gene is essential for function.
Exactly.
So, right.
Exactly. So that's what the neural network is trained to be able to learn and deal with, right? The neural network basically gets expression counts for the 20,000 genes in the case of right, for, let's say, for a human, right? And it is basically told or trained to identify, based on the co-expression patterns of those genes, what kind of essentially constitutes the identity of that cell. So it's really the neural network that is doing that work, and you are not now saying, okay, I'll do this manual gene expression, differential gene expression analysis.
And how
it's a different approach.
And how do those genes that it marks as relevant line up in response to marker genes? Like, do you see all the marker genes represented, or do you see like totally different gene lists when it identifies the cell type?
The method does not identify the cell type. What the method does, it gives you a very compact representation or the embedding for that given cell. The beautiful thing of the method is that it can take any cell from any organism and map it into the same space, with the idea that cells that are mapped closer together are more similar, right? Or that cells that belong to the same cluster, they have the same type, they have the same state, and so on, right?
But does it give you the top list of genes that cause it to map? Like, does it give you like, these are the gene profiles that we use to map these clusters? Does it tell you any of that?
It will not. You can do this on top. What it will allow you to do is to reason about this cell states at a much more finer grained level, beyond at much more finer grained level. So it's more general, it's beyond, right? Of course, you can identify the cluster and then say what genes, are overexpressed and so on. You can do that, but you can do something else as well. So you can always do that type of analysis that you are talking about.
All right, thank you.
Thank you.
And since we're about 5 minutes past time and there's some questions in the Q&A, I would say anyone else with questions, go ahead and put them into the Q&A in Teams. It'd be great if you could go in and answer those, asynchronously as we move on. So thank you again, Dr. Leskovec, for
Thank you so much.
your keynote presentation.